DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long spiraling ladder. DNA is what stores our genetic instructions, the information that programs all of our cells' activities. It's a 6 billion letter code that provides the assembly instructions for everything that you are. In which case, every body cell or somatic cell in you right now has 46 chromosomes, each containing one big DNA molecule. These chromosomes are packed together tightly with proteins in the nucleus of the cell. That DNA acts as a blueprint or recipe for living things. But how? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important they are often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The cool thing about them is that they can be attached to each other like Legos. Legos produce an endless variety of larger particles called proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. These proteins that make up our bodies, and keep in mind, there are millions of different kinds of proteins. They each have to be formed in the perfect shape in order to function. If they are in the wrong shape, they usually won't work. That's where the DNA comes in. But one of its main and most well understood function is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built all at the right time and in the right place, everything else, from cells to organs to entire creatures, will come out just fine. DNA is a nucleic acid, and so is its cousin, which we'll also be talking about, ribonucleic acid or RNA. Well, nucleic acids are the fourth major group of biological molecules, and they have the most complicated job of all. Structurally, they are polymers, which means that each one is made up of many small, repeating molecular units. In DNA, these small units are called nucleotides. Link them together and you have a polynucleotide. Let's take a look at what makes up each nucleotide. There are three things which make up each nucleotide. Number 1, a 5-carbon sugar molecule. Number 2, a phosphate group. And number 3, one of 4 nitrogen bases. DNA gets the first part of its name from our first ingredient, the sugar molecule, which is called deoxyribose. But all the really significant stuff, the genetic coding that makes you you, is found among the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It's important to note that in living organisms, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. They are like an intertwined microscopic double spiral staircase. In DNA, the sugars and the phosphates bond together to form twin backbones. These sugar-phosphate bonds run down each side of the helix, but chemically in opposite directions. In other words, if you look at each of the sugar-phosphate backbones, you'll see that one appears upside down in relation to the other. One strand begins at the top with the first phosphate connected to the sugar molecule's 5th carbon and then ending where the next phosphate would go, with the free end at the sugar's 3rd carbon. This creates a pattern called 5' and 3'. Now on the other strand, it's exactly the opposite. It begins up top with the free end at the sugar's 3rd carbon and the phosphates connect to the sugar's 5th carbon all the way down. And it ends at the bottom with the phosphate. And you've probably figured this out already, but this is called the 3' to the 5' direction. The nitrogenous base is always attached at the 1 carbon of the sugar. If we count from there, we can see there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. These two long chains are linked by the nitrogenous bases via relatively weak hydrogen bonds. One nucleotide has an adenine base, or only thymine can be its counterpart. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. The guanine to cytosine pairing has 3 hydrogen bonds, making it slightly stronger than the adenine to thymine base pair, which has only 2 bonds. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pairs specific information, while the minor groove is largely base pair non-specific. This is because the patterns of hydrogen bond acceptors and donors that proteins can interact within the grooves. In this way, the DNA can be acted upon in either sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. RNA is similar to its cousin DNA. It has sugar phosphate backbone with nucleotide bases attached to it. But there are 3 major differences. RNA is a single-stranded molecule, no double helix here. The sugar in RNA is ribose, which has 1 more oxygen atom than deoxyribose. Also, RNA does not contain thymine. Its 4th nucleotide is the base uracil, so it bonds with adenine instead. RNA is important in the production of our proteins. It has a crucial role in the replication of DNA. Every cell in your body has the copy of the same DNA. It started from an original copy and will copy itself trillions of times over the course of a lifetime, each time using half of the original DNA strand as a template to build a new molecule. Helicase is marvelous, unwinding the double helix at breakneck speeds, slicing open those loose hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication fork, and it has a top strand called the leading strand and another bottom strand called the lagging strand. These unwound sections can now be used as templates to create 2 complementary DNA strands. But the 2 strands go in opposite directions in terms of their chemical structure, which means making a new DNA strand for the leading strand is going to be much easier than the lagging strand. For the leading strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down the molecule. But before it can do that, it needs a section of nucleotides that fill in the section that's just been unzipped. Starting at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer, just a little thing for it to hook on to so that it can start building the new DNA chain. And for that little primer, we can thank the enzyme RNA primase. The leading strand only needs this RNA primer once at the very beginning. Then the DNA polymerase says, "I got this," and just follows the unzipping adding new nucleotides to the new chain continuously, all the way down the molecule. Copying the lagging strand is difficult to understand. This is because DNA polymerase can only copy strands in the 5' to the 3' direction, and the lagging strand is 3' to the 5'. So DNA polymerase can only add new nucleotides to the free 3' end of the primer. So maybe the real problem here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied as a series of segments. Here, that awesome enzyme RNA primase does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backward along the strand. This is done in a ton of individual segments, each 1000 to 2000 base pairs long and each starting with an RNA primer called Okazaki fragments. After these allow the strands to be synthesized in short bursts. Then another kind of DNA polymerase has to go back over and replace all those RNA primers, and then all of the little fragments get joined up by a final enzyme called DNA ligase. DNA replication gets it wrong about 1 in every 10 billion nucleotides. But it turns out DNA polymerase can also proofread, in a sense, removing nucleotides from the end of a strand when they discover a mismatched base. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of the free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Their small shape and size allows them to fit through tiny pores in the nucleus out to the cytoplasm and into the mouth of another particle called a ribosome. Ribosomes are protein building machines. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosomes reads this code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acids. Transfer RNA molecules carry the amino acids to the ribosome. They read the RNA code 3 letters at a time, sucking amino acids out of their surroundings, sticking them together in a chain according to the genetic code. As a chain grows, it bends, folds, and even sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added to the next. For example, CAA tells the ribosome to grab a glutamine, AGU tells it to grab a serine, and so on. Once a protein is built, then it goes on to do a number of different things. This is a 3D model of a DNA, and it has been made on behalf of the entire art project team. As you can see, I have used clay, paint and toothpick for creating this model. This model has 8 pairs containing 2 segments. The red denotes thymine, the green denotes adenine, and the blue denotes guanine, whereas the yellow denotes cytosine. The white backbone of this structure is the sugar phosphate. Thank you. Thank you for being attentive. Stay home, stay safe.